Okay, it's a great delight to be here. Welcome to Harvard. I won't say I hope you come because I know you all will come. And I'm excited to see many of you in Life Sciences 1B, all the budding scientists next year. So what I'd like to do in the next 10 minutes is tell you a little something about what Darwin didn't know. Arguably one of the most profound ideas in the sciences is the idea of evolution by natural selection. Profound because it explains how the great diversity of organisms that we see on our Earth first arose, but also profound in its simple elegance. Darwin's ideas, which he articulated in his 1859 book entitled On the Origin of Species, was a great tome that articulated these three pillars, different than the pillars that Dr. Chetty just mentioned. The idea is, first, if individuals within a population compete, that is, there aren't enough resources to support an infinite population size, therefore individuals will compete to survive and reproduce. Second, if there's variation in their ability to compete, with some individuals being more competitive than others. And third, if the traits that make those individuals more or less competitive are heritable, then that population should evolve by natural selection. Well, in his book, he spent decades providing evidence for competition between individuals, both through observation and careful experimentation. He showed that in many populations, there's variation in individuals' ability to compete for resources and survive and pass on their genes. But the thing that was missing from his theory in 1859 was the idea of heritability. Both Darwin and his colleagues surmised that many traits were heritable based on the similarity of offspring to their parents. But what Darwin didn't know, what was unknowable at the time, because of course this was before we knew about genes and genomes, was exactly how traits were inherited. So next what I want to do is tell you a brief anecdote that links Darwin to that second great discovery of how traits are inherited. So here what you're looking at, I don't expect you to read it, but just appreciate maybe a few oohs and ahs. This is Darwin's last, thank you very much, Darwin's last publication, published just 3 weeks before he died in 1883. And it's published in a preeminent journal called Nature, still a journal that we publish in today. It's entitled On the Dispersal of Freshwater Bivalves. And really what this is a little natural history observation of a specimen of a beetle, and clamped to its leg is a cockle, or a freshwater clam. Why was this published in Nature even a century and a half ago? Well, it turns out this solved this great mystery about how these clams could move from lake to lake in the British Midlands. They could hitchhike on things like beetles that could fly them around. But I digress. The reason that I wanted to tell you about this story is to tell you about how Darwin got his hands on this particular specimen. It was sent to him by an amateur naturalist who lived in northern England, who was a shoemaker by training. He sent the specimen to Darwin. They had a flurry of correspondence. Well, it turns out this young shoemaker's name was Walter Drawbridge Crick. And to those of you who are budding scientists, that should ring a bell, because nearly a century later, this shoemaker's grandson and his buddy, Jim Watson, a former Harvard professor, with ideas either borrowed or stolen, depending on your perspective, from Rosalind Franklin, made this second great discovery, that is the three-dimensional structure of DNA, which explained how that genetic information could be stored and how it could be replicated and passed on from generation to generation, what Jim Watson humbly referred to as the secret of life. And in this genetic code, we find even more evidence for Darwin's great theory. And now, as scientists, we're poised to ask questions like Darwin did. How did this diversity of life first evolve? But now we can look for answers in the genetic code. So today I'm going to tell you a brief story about what we can learn from identifying genes that contribute to differences that matter in individuals' struggle to survive and reproduce. So the main actor in today's story is this little charismatic mini fauna shown here. This is the deer mouse, which is the most abundant mammal and widespread mammal in North America. And if you were interested in going out and catching this mouse, you would go to habitat that looks something like this, where densely vegetated, dark soil, and the mice you would catch would look something like this. A typical mouse, a dark brown coat, a light-colored gray belly, and a nicely striped tail. But about 3,000 years ago, these mice colonized the beautiful white sand beaches of the Gulf Coast of Florida and the Atlantic coast of Florida, where they inhabit these beautiful white sand dunes. And if you go to these areas, maybe at spring break, for example, and you catch a mouse, the same species of mouse would look very different. It looks like this. It lacks pigment on its face, on its flanks, and on its tail, and only a tan stripe on its back. Now, many of you, I'm sure, are thinking, well, this makes sense, right? They're camouflaged in their environment and probably can hide from all those things that want to eat these poor little mice. Well, that's a great hypothesis, but as scientists, we want to test those hypotheses with experiments. And that's exactly what a Harvard undergraduate who was working in my lab did. She set out around February, when it's awful and cold here, to the beautiful beaches of Florida. And there, she armed herself with hundreds of models of mice. And those models of mice, she either painted dark brown or white with a tan stripe. And she set them out at 2 field sites, one on the beaches, one in the mainland. She set them out randomly in equal frequency and came back the next morning to see how many were attacked by visually hunting predators like owls, herons, hawks, or foxes and coyotes. And what she found, quite to our delight, was exactly what we thought. That is, mice that matched their habitat, like this one here, or these 2 top panels, had many fewer attacks than those that were mismatched. So this experiment clearly demonstrated that color matters for survival in these mice in their natural habitats. But next, what we wanted to know is something about the genetic basis of this difference. So we moved from the field back into the lab. And over the next several years, we interrogated the nearly 3 billion base pairs in the mouse genome, which was comprised of about 20,000 genes, to hone in onto one gene in particular, the melanocortin-1 receptor. This particular gene, when we looked at it in dark mice versus light mice, we found that there was one little change, a single DNA base pair change that encoded an amino acid change in this receptor that changed the function of that receptor that then changed the color of the mice. So in other words, we were able to identify a genetic mutation, understand how it affected the proteins, how it gave rise then through development to this color variation, and ultimately how this affected the ability of the mice to survive in their natural habitats, thereby illustrating the full picture of evolution by natural selection. And we now use this model as a teaching tool in Evolution and Life Sciences 1B, where we can actually simulate mice running around on dark soil, getting picked off by these hawks, and or on light soil, and then we can track how the different alleles or different versions of this melanocortin-1 receptor change over evolutionary time. But these beach mice still had a little more to tell us. One day I was at a conference sitting next to a colleague of mine who was working at the University of Leipzig, and he had just sequenced the entire genome from woolly mammoths. He had 3 individuals where he could get DNA from bones that were frozen in the Siberian permafrost. We're sitting next to each other, and I said, let's look at the melanocortin-1 receptor. He said, sure. So he got onto his computer, pulled up the gene, and what he found, quite to our surprise, was that the same gene, there was the same mutation, which suggests that woolly mammoths may have been polymorphic in color as well. And it's not just mammoths. It turns out that different mutations in the same gene lead to differences in protein function, differences in color, and differences in survival. Like these white lizards that live on the white sands in New Mexico compared to their dark ancestors. For those of you who have a Labrador retriever at home, whether you have a golden lab or a black lab, a single mutation in the melanocortin-1 receptor. For those of you that have reddish hair and pale skin like me, you may also carry a variant of the melanocortin-1 receptor. And it's not just us. It's our close ancestors or cousins as well. About 6% of Neanderthals carry a variant of the melanocortin-1 receptor, and hence we think about 10% of Neanderthals also were red-headed. So it's fun now to think about how tickled Darwin would be, not only that his process of evolution by natural selection, that missing piece has been discovered, but by studying the genes that give rise to natural variation, we can learn things not just about beach mice and mammoths and lizards and how they survive, but about ourselves, our pets, and our close cousins. Thank you very much.